the slide
Yes.
scroll.
Yeah, I'm still waiting for it. Okay, here we go.
Yeah, there now. It's the real one.
Yeah, laser pointer.
It's a different first slide.
It is due to recent events. You'll see in a second.
Okay.
Add some animations and such.
Yeah, there now.
Okay, you're live.
Yeah, I see that we are live. Okay, we'll start in a few minutes, everyone. Okay, it's now 5 o'clock. So I don't think my camera is working, but you can hear me. My name is Anna Vlasits, and I'm a postdoc in Thomas Euler's lab. And I want to welcome you to this session of the Tübingen Neuro Campus seminar series. Thank you all for coming today, and apologies for the technical difficulties with the talk last week. Today, I am very pleased to introduce today's speaker, Professor Alon Poleg-Polsky. Professor Poleg-Polsky cut his teeth as a neuroscientist in Jackie Schiller's lab at the Technion in Israel. There he began his career-spanning work to understand how dendrites perform computations. Dendrites are the input structures of neurons, where neurotransmitter receptors convert chemical signals to electrical signals. One major question in neuroscience is how different properties of dendrites contribute to the information that is integrated and passed from neuron to neuron within neuronal circuits. During his MD PhD work in the Schiller lab, Professor Poleg-Polsky studied pyramidal neurons in the cortex, showing how thin dendrites and NMDA receptors are involved in nonlinear processing of information. Professor Poleg-Polsky then turned to the retina, performing postdoctoral research in Jeffrey Diamond's lab at the NIH. There he applied his expertise in dendritic integration to questions of visual processing in the retinal circuit. In particular, he studied the role of dendritic integration in motion processing in the direction selective circuit, and showed that ganglion cells and amacrine cells rely on specific dendritic properties to transmit motion information reliably. Now, in his own lab at the University of Colorado in the Department of Physiology and Biophysics, Professor Poleg-Polsky has continued his work on retinal motion processing using electrophysiology, two-photon imaging, and computational modeling methods. Today, he will tell us about his most recent study titled Novel Object Detection and Multiplexed Motion Representation in Retinal Bipolar Cells. Welcome, Professor Poleg-Polsky.
Thank you, Anna, for this very nice introduction. Yes, indeed. Today I'm going to talk about motion processing and novel object detection in retinal bipolar cells. And I hope that this talk would go better than last time when we experienced some technical difficulties and even got to meet Zoom tools. Yes. So actually, the reason I put this animation here is to show you an example of what we mean by novel objects, objects that suddenly appear in the scene. And in my talk today, I'm going to start with describing our recent findings of the difference between motion and stationary processing in bipolar cells. I then go and tell you about how motion processing depends on the presence of static occluders or edges. And in the second part of the talk, I will show you that our findings probably represent a previously unappreciated ability of center surround receptive fields to identify novel objects. And last, I will show you that we think that these types of computations begin very early, possibly even in the photoreceptors. Okay, and I'll start with a brief introduction to bipolar cells, which are the star of today's talk. So here you can see a schematic of the circuits that bipolar cells are embedded in the retina. They receive input from photoreceptors and provide output to postsynaptic cells like ganglion cells, which form the output of the retina. And in between, they're innervated by horizontal cells and amacrine cells. These circuits also form the receptive field properties of bipolar cells. And bipolar cells have very simple and common receptive field, which is called center surround receptive field organization. And in general, receptive field means the area in sensory space, visual space in this case, to which the cell responds to. The center component of bipolar cell receptive field is formed predominantly by photoreceptors, and it's excitatory. The surround component of the receptive field is formed by lateral inhibition coming from horizontal cells and amacrine cells. So just keep this information in mind. We'll return to receptive field properties towards the second half of the talk. There are about 15 types of bipolar cells in the mouse retina, which is less than the number of ganglion cells, which is about 40 types, and amacrine cells, which maybe between 50 or 60 types. And also bipolar cells convey all the visual information coming from the photoreceptors, which in principle allows us to record and see the entire representation of visual information in specific cell population. And this could be done as in a very clever experiment performed by Catherine Franke, Philip Berns, Thomas Euler, Tom Baden and colleagues. They expressed iGlu sniffer, which is a glutamate sensor, in postsynaptic cells so that they can see release from bipolar cells. And one piece of information that I didn't tell you is that the axonal stratification of different bipolar cell types, shown here, is different and depends or is in different depth in the inner plexiform layer of the retina. And so what the researchers did here was to record activity or glutamate release in response to visual stimuli, shown schematically here. And then they grouped different regions with different or similar response property into clusters, which then they sorted based on depth in the retina. And this corresponds to the release of different bipolar cell types. From this figure, you can also see the general principles of organization of bipolar cells. The deeper bipolar cells, or more precise bipolar cells that have axons that stratify deeper in the inner plexiform layer, they're typically or respond to light offset, so they're off bipolar cells. More superficial cells are on bipolar cells. And also you can see that in terms of a release, dynamics of the release, cells in the middle of the IPL, inner plexiform layer, tend to be more transient. Now, this difference in release from bipolar cells is thought to be important for processing in the postsynaptic circuits. And to give you one example, is recent, I would say, pretty recent idea of how differential release from bipolar cells can correspond to very interesting computation of direction selectivity. And the idea is the following. If you have a postsynaptic cell, as depicted here, that is innervated in different regions by different bipolar cells, which have different release dynamics. For example, this cell number 1 more sustained release, this cell number 2 has more transient release. If you activate these bipolar cells in different sequences, it turns out that some of these activation patterns would elicit larger postsynaptic responses. And this is loosely based on the Reichert type detector. I'm not going to go into the exact mechanism of this. This is not important for our presentation, but this idea was very fruitful in the field. It was first described by Sebastian Xong group, who showed this type of anatomical arrangement of different bipolar cells on to starburst cells dendrites. And this was followed by different groups. More recently, similar spatial arrangement was found by Jon Harlab in collaboration with Kevin Brigman. But what is important for the discussion that we are having today is that when bipolar cell release was measured, it was typically investigated using non-moving stimuli, such as full field flashes. But the computation that is done here is direction selectivity, which relies on moving objects. And so in the first experiment that we did, we asked this very simple question. Is the representation of stationary object similar to representation of moving objects in the proportional population? Okay. To do so, John Gaines, who is a postdoc in the lab, and he did most of the experiments I'm about to show you, he followed the recipe developed by Catherine and Philip. He introduced iglosniffer to the retina using viral vectors, waited for good expression, dissected the retina, placed it under a microscope dish where you can stimulate with different patterns, and record the fluorescent activity using two-photon microscope. And for this first experiment, John stimulated the retina with two types of stimuli: stationary flashes, simply turning lights on, or moving bars. So the animation didn't work, but yeah, moving bar. And here I want to show you example responses to moving bar. This is raw fluorescence, and this is change in fluorescence from such a release of glutamate. And he would image the retina. He would do it in different imaging planes, so we can see the responses across the IPL. You'll then extract all the active pixels. Here's an example showing pixel activity. The color coding indicates change in fluorescence for stationary stimulation for moving bar. And then we would combine pixels with similar response properties into regions of interest. Here in this, for example, you can see here is the region of interest that is responding off type. You can see begins the response after the flash. Here's a transient on response, more sustained on responses. And so this is done in a single experiment. We will combine such ROIs from multiple experiments, and John did more than 100 retinas. And we will combine these ROIs into functional clusters of activity, very similar to what Catherine and Philip did. The clustering was done strictly based on the responses to stationary flashes. And our results basically, I would say, replicate previous findings. With more with off responses being deeper in the retina. Here on this plot, you can see the depths in the IPL, and more on responses being more superficial, and again, more transient clusters in the middle of the IPL. So this is, again, this is stationary responses to bright flashes, dark flashes. And now I'll show you the responses to moving bars. And similarly, the first column here shows responses to bright moving bars, and the second column shows responses to dark moving bars. And what we see is quite a lot of diversity in responses to motion. Some clusters, let's say this cluster number 2, the shape of the response to motion is very similar to the shape of the response to the stationary flash. In other clusters, like let's say cluster number 6, the responses to motion are much smaller than the responses to stationary flash. And you can see that many clusters, that they are also more sluggish, at least in the rise time. And actually, to see this better, let me just focus on the rise time, like the first half second or so of the response. And once again, I show you the responses to stationary stimulation versus responses to moving bars. And what you can see, and this is very evident for the on clusters, is that whereas the responses to the stationary flash have very similar rise times, the responses to motion from the corresponding clusters are very different. And we can analyze this as following by plotting the rise time of the motion responses versus the rise time of the stationary responses. And if you can see, this is the trend line. There is no correlation between the rise time of the stationary flash and the motion response. If you analyze the decay time, we can see it's more correlated, but still the correlation is not perfect. So from this, we deduce that the responses to motion cannot be predicted strictly based on the shape of the stationary response. But what can predict them? So we considered two options. First, we thought maybe the responses to motion differ based on the depth in the retina. And the second option that we considered was perhaps the responses to motion depends on other properties of bipolar cells. And the most obvious one was the kinetic release, how the transient was sustained. To explore these options, we performed the following analysis. First, we computed the ratio between the stationary and the motion responses. Here I show you the ratio between the peaks. And what this means is, let's take cluster number 6, for example. The stationary response was about twice as large as the motion response. And for cluster number 2, this guy, the responses were similar. And then we simply looked at them as a function of cluster number, because you remember, clusters are sorted based on their depth. So this would give us a glimpse on the relationship between the motion responses and depth in the retina. And here, we don't see any clear relationship. For the second analysis, we computed the transiency index, which is about 1 for very transient clusters, like cluster number 6, and closer to 0 to very sustained clusters. And once again, we plotted the transiency index versus this ratio between the stationary and motion responses. And now, we observed a very nice and clear correlation with the more transient clusters being much more affected by motion, being, in this case, the peak reduced by moving stimulus. We did the very similar analysis for the dynamics, and I've started with decay time. Once again, there was no correlation between decay time and depth in the retina, but a nice and clear correlation with transiency. And the more transient clusters, more affected by motion, they become sluggish with motion. For the rise time, it's quite the opposite. No clear correlation with transiency, but it seems like the more superficial clusters are more affected by motion. In this case, again, the ratio indicates that the responses to motion are sluggish. So for these clusters, the responses to motions were up to 6-fold slower. So what we can conclude from this set of experiments is that motion responses cannot be predicted based on the shape of the stationary response alone, then that we need some other another information, like depth in the retina or transiency of the response. Okay. In the second set of experiments, we decided to investigate the dependence of motion on the presence of edges or occluders. And to understand why we did those experiments, let's talk a bit about center surround receptive fields. And what I'm going to show you now is, I would say, introduction to neuroscience, textbook level of description. What are center surround receptive fields? So let's imagine for a second that this is our visual space into which we can present visual stimuli, such as this dotted square. And this visual space is viewed by cells, such as those, that have center surround organization, as I showed you before. Those cells can be in the retina or other brain regions that have center surround organization, such as thalamus or V1. And what we know, based on this simple model of receptive field, is that this receptive field would respond to stimulus if it lies within their receptive field area. And now I'm going to talk about the functional properties of center surround receptive fields and describe the well-known fact that they enhance representation of edges. And to do so, I'm going to focus on these 3 cells, just for example. We're going to present a stationary flash. This is all a model, very conceptual, theoretical model. And we're going to start with this middle cell, which is activated by the stimulus over its entire receptive field. So in this case, shown here, we can plot the activation of the receptive field components. Here's the center, and here's the surround. And let's say that the stimulus begins here, and at this point, this is just a model, made-up model. And by the way, I'm showing the surround in this orientation, but actually it's antagonistic. It should go down. When we compare the activation of this cell to a cell that lies near the boundary, so this corresponds to this left cell, and that cell corresponds to this right cell, what we see is that activation of the center is roughly the same. That's because I placed the cell, so it will be activated in the center. But the engagement of the surround is reduced near the edge. And that's simply because part of the surround is not activated by stimulus for this part. If we go then and compute the full receptive field activation, and here, very simply, I combined the center and surround, you'll see that the cells near the edge would have larger, maybe slightly larger activation of the receptive field. You can see the difference here in the peak and in this sustained phase. And so this is a well-known fact that center surround receptive fields care about edges. And so we wanted to ask, well, here we showed you how they respond to stationary flashes, but what happens when you present a moving signal? So to explore this, we decided to show our regions of interest edges. And to do so, we started with our original stimulus. In this case, this would be the full field stimulus. And then we simply masked half of the display. So here's a mask. And what this creates is an edge or boundary between the mask and the stimulus. And we can record responses from regions of interest that lie near this edge. Here's an example. Here I show you two responses from transient regions of interest and sustained regions of interest. And the reason that I show you both is that as I showed you before, motion dynamics depend on transiency of the responses. This trace represents a response to full field stimulus. This is response over time, fluorescence over time. This arrow indicates the horizontal position of the region of interest. Here, the response from the same ROI in the presence of edges. So you see it's very similar to a conceptual model, slightly larger. Here's the same from sustained cells. So this was stationary stimulation. For the stimulation with moving bars, once again, we started full field stimulation as before, presenting the same mask. But now, actually, we have two types of responses. One is motion from an edge, or from the mask. And another is motion towards the edge, to this mask. So we decided to show all these stimuli to ourselves. And this is what we got. Once again, the stationary responses that I showed you before. Responses to full field motion. This is exactly the same as in our first experiment. Smaller responses, more sluggish. They don't have direction selectivity. They respond to all directions in the same way. And now responses to motion in the presence of edges. And here we have a surprising result. What we see is that motion from an edge is much larger than motion towards an edge. In fact, motion towards an edge looks very similar to full field motion. Let me summarize these results and start with the outcome of something like our first experiment. So what I'm showing you now are the normalized peak responses. They're normalized by full field motion, exactly as we did for the ratio in the analysis for the first experiment. And let me add the data points. So here you can see responses to edges, or stationary flashes, the difference between the edge response and the full field response. This is the well-known edge enhancement of center surround receptive fields. Now we can see very clearly that motion responses are very different near edges, and motion from an edge is much larger than full field motion or motion towards an edge. And similarly, when we examine the dynamics of the responses, motion from an edge was different from full field motion or motion towards an edge, and it was, I would say, in between full field stationary responses and full field motion. Here, I would like to stress that we are not the only ones who found this result. In a recent paper by Thomas, Anna, and colleagues, they did a similar experiment where they introduced a sniffer and recorded responses from IPL, and they found very similar results. Responses from an edge, shown in black, are larger than responses to an edge, shown in gray. And so, to conclude this part, what our results and our colleagues' results show is that bipolar cell responses depend on edges, particularly so for motion stimuli. What I want to show you in the next part of the talk is that we think that These results represent previously unappreciated property of centers around receptive fields to detect novel objects. And to show you what I mean, I'm going to change some of the labels on our experiment. So I showed you this plot on the previous slide, responses to stationary stimulation, and normalized by responses to motion. And I show you in the bottom some example traces. What I'm going to change is the label of this stationary response. I'm going to flip it and say, well, actually, this is a flash. This is sudden appearance of a new object. In this case, this bright spot. I'm also going to change this insert representing motion to an edge. And I'm going to flip it around, just like that. This is a symmetric representation. But the reason I'm doing this is that now this scheme of different motion responses corresponds to a natural sequence of events happening for moving stimulus in the presence of occluders. And let me explain what I mean. Let's say that we have a window like this, from which we can see the visual scene. And let's say that there is a moving object that moves across the scene, like that. Okay, we saw this guy before. We can break this motion into sequence of events. First, this moving object enters the visual scene from an edge, from an occluder. It continues to move, we call this established motion, and eventually it exits the visual scene, going towards an edge. What I want to claim is that our experimental recordings correspond to these sequences. Motion from an edge is really object entrance. Full-field motion is similar to established motion, and motion towards an edge is similar to object exit. Now you can say, well, mice don't have windows, like they. But actually, any natural occluder would do. So this is an example of a very different type of occluders, and still we have this sequence of events. So let me change the labels again, one last time. And I would like to say that sudden flashes and object entrance to the scene correspond to novel objects. Whereas motion over the full field or towards the edge is pre-existing motion that can be predicted from previous time points. And now we can see how the responses in bipolar cells are different. They're larger for novel objects and categorically smaller for pre-existing motion. This, by the way, these types of novel objects are not the only possibilities for how we can, for how objects can appear in the visual scene. And our colleagues showed that bipolar cell responses are also larger for a different type of novel object. In this case, a looming object, so an object that appears in the visual scene and expands towards you, like this. So what they showed, here shown in black trace, that responses to such objects are larger than responses to the opposite type of object, a receding object, an object that disappears into a point. And once again, looming object corresponds to novel object, and receding object is pre-existing object that disappears. Right. So so far what I told you was semantics. Right? I just changed the labels. But now I would like to show you the model that we think explains these findings. And I would like to circle back to this very simple representation of centers around receptive fields, which I showed you before, in which we presented first a stationary flash and observed the responses from these three cells. Now let me replace the stationary flash with a moving bar, and I constrain this bar so it only appears in this window. This would create edges. This would, because the bar is going to move from left to right, this cell on the left would experience motion from an edge, this cell in the middle would experience full field motion, and the cell on the right would experience motion towards an edge. This is a stimulus. So let's analyze now the responses of the receptive field. And again, we're talking about linear responses from just based on the engagement of the stimulus with the receptive field. Again, we're going to start with a full field response. And what is pretty much obvious is that when the object enters the receptive field, it always enters the surround first. And you can see this here. These traces show you, again, evolution of receptive field components over time. And you can see that the surround starts earlier. This primes the surround to be more effective in inhibiting the center. Same thing happens for this cell that experiences motion towards an edge. This is this cell. Once again, the stimulus enters the receptive field surround. And we call this effect preceding inhibition. Inhibition that precedes the activation of the center. What about the cell that experiences motion from an edge? So in this case, there is no preceding inhibition, and that's because the center and the surround are activated at the same time. The area of the surround that should have been causing the preceding inhibition is not responding. This is this part, because it's not engaged by the stimulus. So therefore, without preceding inhibition, the full receptive field is larger from this cell, which experiences motion from an edge. In comparison now, if we put the response as stationary flashes, again, this is in this model, what we see is that preceding inhibition occurs for pre-existing objects or pre-existing motion. And when it's absent, the responses become larger. And these responses correspond to appearance of novel objects. So just to give you a summary of this part, is that we show using very simple linear model of center surround receptive field that we think that center surround components can enhance representation of novel objects. And once again, this all relies on known properties of receptive fields. And we think, by the way, that this should apply not just to vapor cells, but to any cells that have such center surround organization. Now, in fact, our own visual system heavily relies on center surround receptive fields. And so it's likely that we should experience these effects ourselves. And this is, I would say, is in line with psychophysical studies, which show the different salience of visual responses. So typically, we think that motion is salient. It's an attention grabber. But in fact, what is known from psychophysical studies is that pre-existing motion, usually old motion, is not salient. And the reason is that in many cases, motion is created by our own locomotion in the environment. And so if we'll be paying attention to it, we'll be distracted when we move. What psychophysical studies show is that the most salient stimulus is a sudden appearance of a new object, just as we showed in the proposals. And it's more salient than novel motion being pre-existing object that begins to move. And the explanation is that this type of visual stimuli are much more likely to represent animate objects. So it's important for us to pay attention to those. And by the way, if we plot our experimental stimuli, so we present novel objects, we show strong vapor responses, we show weak vapor responses to old motion. I would say that object entrance in our case is in between novel object and novel motion, and object exit is similar to pre-existing motion. Okay, in the next slide, I'm going to try something, and I hope that it works. And this would be something like a test to see this effect. So what I'm going to show you are going to be 4 squares. The top 2 will begin to move when I click on the slide. Eventually, they'll become old motion. At some point after a few seconds, this square will begin to move. This will be new motion. and then a square, which is not shown here, but it would appear in this place. And now, based on our experimental results, we can make predictions on how these objects will be perceived. So if we look on the bipolar responses, we see that new objects have larger response amplitude. And in our visual system, this would correspond to perception of stronger contrast, brighter stimuli. So this new object would appear brighter. In terms of dynamics, the flashes had the fastest responses. So what we predict that this new object would appear sooner on the screen, even though they actually new motion and new object appear at the same time. Okay, so let's run it and see if it works. Old motion. Okay, I hope this works on Zoom, but let's do it again. Pay attention, it will be brighter and sooner. You can let me know later on if it worked. But anyhow, what I wanted to show you here is our perception of similar objects. And the above-mentioned psychophysical studies, when they investigated the origin of this difference in perception, they concluded that based on the latency, it has to begin in the retina. And I would say that our experiments show a possible explanation when they happen. Okay. Now, in the last part of my talk, I would like to dive in a little bit into the circuits that mediate motion computations in bipolar cells. And I would start with discussion of the circuits that mediate the surround of the bipolar cell. Again, this is a slide I showed you before. And the reason that I want to focus on the surround is that, as I showed you, this preceding inhibition formed by the surround is vital for motion processing. So once again, surrounding bipolar cells is generated by horizontal cells and endocrine cells. And if we focus in on the actual synaptic elements, it is known that direct inhibition onto bipolar cells is mediated by GABAergic and glycinergic receptors. But there is an additional pathway by which horizontal cells can inhibit photoreceptors. Let's call it the Fabry pathway. That is not dependent on GABA and glycine receptors. And so these properties of the circuits allow us to use pharmacology to try to dissect the different components that contribute to the surround. So what we did, we recorded the responses in control condition. This is shown in black for full field. Here's full field stationary stimulus, full field motion, and then we applied blockers of GABA and glycine receptors. And the traces after the blockages are shown in blue. And what you can see is this blockage changes the shape of the response in this example to stationary stimulus slightly, and it didn't affect the responses to motion. Here is a summary plot of the effect on the peak of the response. The blockers are again in blue, and you can see that they didn't affect responses to motion that much. And similar findings were found for the dynamics of the response. I'm not showing them here. And what this means is that we can rule out amacrine cell inhibition and direct input from horizontal cells as mediators of the surround, which leaves only 3 players that contribute to motion processing. And these are photoreceptors, horizontal cells that inhibit photoreceptors directly, but don't inhibit bipolar cells. And the bipolar cells in this scheme receive input directly from the photoreceptors. And that's it. This is sufficient to create the motion responses. But this finding actually caused us to scratch our heads. Because as I showed you in the very first experiment, there is a lot of diversity in bipolar cell responses to motion. Whereas now what I show you is that all bipolar cells essentially get the same input, this photoreceptor input. So how is that possible? How can different bipolar cells respond to the same input with different response properties? To explore this question, we decided to create a model. In this model, we realistically recreated the phototransduction photoreceptors, their release into horizontal cells, and feedback inhibition of the photoreceptors. We then created arrays of such cells, similar to what is happening in the retina. In the simulation, we can present a stimulus or to engage photoreceptors and record from different cells in the network. In this case, I'm going to show you responses from this center photoreceptor and this center horizontal cell. And as before, we're going to start with responses to stationary stimuli, flashes. This plot shows you the response over time in photoreceptors in black and horizontal cells in blue. This is a response to full field stimulation. Here is a response in the presence of edges, as you can see from the diagram. And interestingly, similar to our simple model, we see that the horizontal cell, like the inhibitor surround, is less engaged by the stimulus. It makes sense actually, because part of its receptive field is not activated, and the photoreceptor voltage is larger. Okay, this is the known edge enhancement. We can also simulate responses to motion, and I would start with full field motion. And again, we record from the same center cells. And when we do this, you can see that the horizontal cell is engaged earlier. This is preceding inhibition, and it inhibits the photoreceptor, reduces its peak amplitude. Same thing happens with motion towards an edge. But similar to our experiments, motion from an edge does not recruit the horizontal cell earlier, and the photoreceptor voltage is larger. So to conclude, we think, based on this simulation, that novel object detection and motion computations begin as early as photoreceptors. Okay, but what about bipolar cells? The reason I did the simulation was to tell you how different bipolar cells can sample the same input and create different responses. So we added bipolar cells to the simulation, and I'm going to show you responses from two types of bipolar cells: transient cell, shown in green, and more sustained cell, shown in red. And their position is shown schematically on top. They're sampled from the same photoreceptors. And what you can see is that, similar to our experiments, this transient bipolar cell prefers novel objects, so its response is larger to novel objects and is diminished for pre-existing motion. Whereas the more sustained cell, its responses are also affected by motion, but not as much. Okay. So that means that we can now explore the mechanism for the difference between the computations in transient and sustained bipolar cells. Actually, I see that I'm running out of time, so I'll make it brief. The explanation for this process relies on nonlinear properties of the receptive field. And very, very briefly, if different bipolar cells have different thresholds of activation, so for example, if a bipolar cell requires larger voltage change in the photoreceptor, larger signal change in the photoreceptor to respond, being more rectifying, if you will, such a bipolar cell would more likely represent the initial component of photoreceptor response, which is typically more transient, and therefore it would inherit the transient shape. It is also going to be more sensitive to small fluctuations in the peak of the photoreceptor response. And so it could amplify responses to novel objects and be less responsive to pre-existing motion. Cells that are less nonlinear, like this guy, would tend to replicate the photoreceptor profile and therefore would be less sensitive to motion. And by the way, these findings or results are very similar to recent description by Helen Schreier and Tim Golish of the difference between nonlinearity of transient and sustained bipolar cells in the salamander retina. So what they showed is that more transient cells have more linear receptive field properties, such as responses to contrasts or spatial differences in the receptive field. And our simulations predict that we can add now a motion processing to this list of the difference between transient and sustained bipolar cells. And we think it's not just a correlation, but this is actually a mechanism that would tend to create linear cells. They would also be much likely to be more transient. Okay, this I would like to end. And just to summarize what I presented, we started this talk with describing the difference in motion processing across all bipolar cell types. I then showed you that motion responses depend on the presence of edges in bipolar cells. And we think that this represents computation that is done in center surround receptive fields that is used to detect novel objects. Our simulations predict, pharmacology predicts, that this computation begins in the photoreceptors and is diversified by the nonlinear properties of bipolar cells. Okay, with this I would like to end. I would like to thank my lab members, and especially John, who did most of the experiments. I would like to thank our funding agency and you for listening. I will take any questions. Thank you very much.
Thanks very much, Alon. Fantastic talk. I really enjoyed that. So let's see, I'm going to start with the first question from Jeff Diamond. He asked, he said, the visual effect, the new object versus new motion, worked quite well for me. Is there some way to confirm that the effects reflect processing in the retina as opposed to higher visual areas?
Right. So this was mentioned in the papers that explore this more like in the passive. They say that if you measure the latency of the responses, difference in the latency of flashes or moving objects, they are so fast that they have to originate in the retina. I didn't find it particularly convincing, to be frank. But I think that, well, it's very difficult to link human perception to actual circuit. And I don't think we can do knockouts.
Yeah. Okay, and so then Thomas had a question. Where did it go? Thomas had a question, Thomas Euler had a question about novel object detection versus contrast encoding. So one of the classic things that center surround receptive fields are supposed to be involved in is contrast encoding. And the question is, what do you see as the essential difference between these two? I mean, how do you see this phenomenon that you're describing as different from contrast encoding?
Well, the stimuli that we presented had the same contrast. And this is very important. Otherwise, we won't be able to tell much. And by the way, for example, the motion towards an edge and motion from an edge, if you count the photons that are emitted from the display, they're the same. They're symmetric stimuli. But I think what is happening is this is going to be confusing. It is for the like the test that I showed. Novel objects appear to be brighter. And I think this is maybe how the visual system works. It emphasizes them, but not just by putting our attention to it, but also making them more visible.
Okay, so then I have a question about the relationship between transient sustained and motion sensitivity. So sort of a naive instinct that I might have had about which of the two, transient or sustained, would be better at motion detection is that maybe a sustained cell would be better because its response lasts longer and could be more influenced by surround inhibition or something like this. So can you revisit the way that you see the relationship between transient sustained and motion encoding?
This is a very good question. Let me go back a few slides. Because what we focused on, let's talk about the simulation. So what we focused on was the difference between new objects, the flashes, and motion. And I told you that, well, transient cells are more sensitive to motion because they are suppressed by motion. Actually, if you want to perceive motion, you want to use these transient cells. And this is a very interesting question because now we should ask this question in terms of postsynaptic computations that go back to the models that were developed of starburst amyloid cells and Diaz ganglion cells showing like this Eicher type direction selectivity. Now that we know that it's not just the shape of the response that's changing, it's the amplitude of the response. And they cannot be really predicted from those stationary stimulation. I don't know. I frankly don't know how this would react. How this all combines together. I think it would change for every postsynaptic cell.
Interesting. Okay. Okay, we have a question from Chase Helmer. He said, really interesting talk. Do you invert the stimulus for on versus off bipolar cells? If so, is the effect of symmetric or asymmetric in on or off pathways?
That's an excellent question. We didn't see an effect on, we didn't see difference between on and off bipolar cells. But I would say that we didn't study it enough. For the experiments I showed you, like this, where we presented edges, we did it only for bright flashes. So I don't know how would off cell respond to a dark stimulus. But when we presented this bright stimuli, the off cells, off bipolar cells, had the same type of response. The response was much larger for motion, form, and edge.
Okay. So it looks like Marla Feller had a similar question about whether this also happens in off cells, but it sounds like you mostly answered that. So the next question was from Simon Laughlin. Were your model photoreceptors nonlinear? Real photoreceptors definitely are when edges have high contrast. And in the housefly, this helps to enhance the response to small moving targets.
And what I missed the last part. What helps?
He said real photoreceptors are definitely nonlinear when edges have high contrast. And he said that in the housefly, this helps to enhance the response to small moving objects.
So the answer is yes and no. The responses were not linear. And I would say they are realistic, but we didn't use full description of the photoreceptor cascade. So maybe halfway.
Mm-hmm. Okay, so then Wan Yulei from John Dem's lab said she had a couple of questions. For the 2-photon imaging, the scanning laser will actually move in the center receptive field and then move out of the surround receptive field. Will this effect entangle with stimulus evoked responses?
So this is an excellent question because I think this is a big problem in our field that our laser can also evoke visual responses. And I didn't tell you much about the stimulation protocol, the recording protocol. So first, we used a photopic regime to try to minimize the effect of the laser. And we have this combination of 2-photon microscopy and confocal microscopy, where we can present the stimulus at the same time as we scan. So we don't need to interleave or use very low stimulus light intensities. The scanning itself is fast. So one line takes us about half a millisecond. And the frame rate of our recording is 50 hertz, so every 20 milliseconds, full frame. This shouldn't introduce activation of the bipolar cell because the scanning also goes from top to bottom, whereas our stimuli move from left to right. And these are very different time scales, 20 milliseconds overall. And the laser's light stays in the receptive field for probably 10 milliseconds or 5 milliseconds.
What was the velocity of your moving stimuli?
The moving stimulus was moving at 0.5 millimeters per second.
Okay, a second question from 1U about the center surround receptive field. Why is the surround receptive field more negative at the center? According to the spatial organization of lateral inhibition, shouldn't it be stronger in the surround and weak at the center?
I'm not sure that I understand this question.
I think it was related to the shape of your surround, like in your modeling, and the shape of the surround, the spatial shape of the surround. That it doesn't have like a trough in the middle or something. I think that's what she was asking.
Oh, okay. So I think what the confusion is that usually on this plot, people also show the full receptive field, which is a combination of the center and surround. And I didn't show it just because I wanted to focus on the subcomponents. But yes, so what you would see if you combine the center and surround is a trace that goes down and then goes up, and goes up where the center is. But this is where we combine them. Right? They combine the two components. This full receptive field. I hope I answered the question. If not, we can talk about it again.
Yeah, I think we'll have a Zoom session afterwards. So if that didn't answer your question 1U, we can discuss this some more. Okay, so it looks like we have another question from Tim Golish. He said, great talk. Could different dynamics of glutamate receptors of bipolar cells contribute to differences in motion encoding between transient and sustained bipolar cells?
Yes, based on our models, definitely. But this won't explain these nonlinear properties. So when we started doing the simulations, I assumed that the difference between transient and sustained bipolar cells would be simply because of the dynamics of glutamate receptors, let's say. And this is true. This works. So if you have a faster responding cell, it would be more transient. Or at least it would have higher rise time. But we found that in those cases, those more transient bipolar cells would have more responses that are more similar to sustained cell. So what I mean is that you can take this sustained cell, and if you make its responses faster, it would become more transient, but it would still have the same motion sensitivity. Only when you consider that the nonlinearities, this rectifying function I mentioned, then it would explain how cells would be sensitive to motion.
Great. If there are no more questions, I think, yep, that was the last one. So now we will move into the offline, or it's still online, the off live discussion of this very interesting work. So please check the chat for the Zoom link, and feel free to join us for some more informal discussion. Thank you very much, Professor Poleg-Polsky, for giving the talk today.
Thank you, Anna and Thomas, for inviting me.